Good afternoon, everybody. Welcome to today's webinar. My name is Chris Harrold. I'm the Yara product manager for the UK and Ireland, and I'm joined today by my colleague Antonis Angeletakis, who is Yara's director of biostimulants. Today's webinar, as the title says, is an introduction to biostimulants, but biostimulants are a large subject in their own right, a complex subject, and we won't be able to cover everything there is to talk about with biostimulants in a 1-hour webinar today. So our plan is to follow up today's webinar in about 10 days' time with a second one. And in that one, we'll build on today's webinar and go into more detail on how Yara itself has been developing its biostimulants for specific crops and markets around the world, as well as here in the UK and Ireland. So I'll be just giving you an introduction to why we're talking about biostimulants today, why they're becoming increasingly important in crop production around the world, and a brief overview of the different types of biostimulants. And as I said at the start, then I'll hand over to Antonis, and he will continue that discussion really around the development, formulation, and how they get used in and validated in trials. Okay. So why biostimulants? Well, farmers around the world, we are being looked at to produce significantly more food to feed an increasing population. 50% more food by the middle of this century is what's talked about. But at the same time as that, they're being asked to do that with a closer eye to farming sustainably. And that means more focus on getting the most out of the inputs that they're using, so talking about nutrient use efficiency, and obviously respecting the environment that we're producing that food in. Greenhouse gas emissions and carbon are obviously key points with all of that as well. And at the same time, there's the challenge around extremes of weather. We see it now more regularly, these either extremely wet periods of weather here in the UK or extremely dry periods as well. And that's all having an impact on how well or how successful our crop production is. And biostimulants are going to play their part in how we go forward in maintaining that productivity, but also having that focus on sustainability at the same time. So what are biostimulants? Well, the international standard definition of a biostimulant is a substance, microorganism, or a mixture that, when applied to seeds, plants, or the rhizosphere, soil, or other growth media, act to support a plant's natural processes independently of the biostimulant's nutrient content. The plant biostimulant thereby improves nutrient availability, uptake or use efficiency, tolerance to abiotic stress, and consequent growth, development, quality, or yield. That's quite a wordy definition, but as I said at the start, the whole area around biostimulants is very complex, and hopefully this webinar will help us to understand that a little bit more. Environmental stresses. Plants are subjected to a wide range of environmental stresses, which can limit the productivity of agricultural crops. And there are two types of environmental stresses encountered by crops. That's abiotic stress and biotic stress. And abiotic stress causes the loss of crop potential worldwide and includes issues around excess water, drought, extremes in temperature, salinity, soil toxicity, etc. Whereas attacks by diseases, pests, nematodes, bacteria, they can be classed as biotic stresses. And are we reaching our maximum yield potential? This was some work looked at by the American Society of Plant Physiologists some time ago now, but you can see here that average yields of a range of crops around the world, highlighted in the blue parts of the bar, are clearly behind where some people are managing to achieve record yields, noted here by the red line at the top of the bars. The biotic stresses that we mentioned, the diseases, the pests, they account for that lighter grey area in the bars. And then there's clearly a big area at the top of the bars that's attributed to something else. What's stopping these crops achieve their genetic potential, if you like? And some of that is clearly down to abiotic stresses. Again, hence the reason that the biostimulants have got their part to play in helping achieve these crops' genetic potential. So biostimulants can have specific benefits. I've already touched on this abiotic stress tolerance, but also they can help with nutrient use efficiency. And that's very important when we know that we're having pressure put upon us as an industry about reducing our carbon footprint. And if we can increase our nutrient use efficiency, that can clearly help with that. And then crop quality, yield and quality. Can we improve the marketable yield of the crops that we're growing, avoiding food waste? And so that's another area where they can help us. So there are different types of biostimulants. The AHDB a few years ago have categorized them into non-microbial and microbial. So in the non-microbial category, we've got things like seaweed extracts, humic substances, chitosan, free amino acids. And then in the microbial category, we've got the bacteria and the fungi, basically. So if we just put a little bit more detail onto those two different categories, starting first with the microbial biostimulants. A microbial plant biostimulant consists of a microorganism or a group of microorganisms. They're living things. Microbial biostimulants include mycorrhizal or non-mycorrhizal fungi, bacterial endosymbionts like rhizobium, and plant growth promoting rhizobacteria. So obviously there are beneficial fungi, but there is a symbiosis between those and plants, and an interaction in plant roots in different ways. Mycorrhizal fungi can establish symbiosis with over 90% of plant species. So they're obviously an important part of this. There's an increasing interest in the use of mycorrhizae to promote sustainable agriculture. That benefit of that symbiosis includes nutrient use efficiency of macro and micronutrients, water balance, and as well as that abiotic stress reduction. There are limitations, though, and it comes around the sort of difficulty in propagating those fungi in a larger scale, because we're talking about putting a living organism into a product. Beneficial bacteria, they interact with plants in many ways, and as for fungi, there are similarities in that mutualism, that symbiotic relationship. Bacteria move from the soil into plant cells by way of the rhizosphere, and these associations may be short-lived or for a lot longer than that, and can be movement from the seed itself. The functions that influence plant growth include supply of nutrients, increase in nutrient use efficiency again, and enhancement of abiotic stress tolerance. So there's a lot of similarities here. Also, similar is the complexity about developing products that contain fungi. The technical difficulties in formulating the inoculants can give rise to inconsistencies in practice. So moving on to what are the non-microbial biostimulants. Non-microbial biostimulants, as is implied by the name, do not contain living microorganisms such as the bacteria or the fungi. And these biostimulants are typically derived from various organic or inorganic substances and can include amino acids, humic substances, seaweed extracts, etc. So just looking at a few of those examples in a bit more detail, seaweed extracts. The use of seaweed as a fertilizer and as a source of organic matter has gone on for thousands of years, but their use as a biostimulant is certainly more recent. They can be applied to soils, used in hydroponics, but also used for foliar applications as well. They have positive effects via the soil microflora, helping growth-promoting bacteria. They also influence positively hormonal effects related to germination, establishment, and further growth and plant development. And anti-stress effects are also reported with protective compounds in the seaweed extracts like antioxidants. Chitosan is derived from chitin, which is an abundant polysaccharide that gives strength to the exoskeletons of insects, crustaceans, and also the cell walls of fungi. Agricultural applications of chitosan have been developed focusing on plant protection against fungal pathogens, but also tolerance to abiotic stress. And this tolerance to abiotic stress is in part due to chitosan's inducing stomatal closure. Humic substances, they're natural constituents of the soil organic matter, resulting from the decomposition of plant, animal, and microbial residues. They can be extracted from naturally humified organic matter, such as in a compost, or from mineral deposits like leonidite, which is a form of lignite. These humic substances are part of a complex connection between organic matter, microbes, and the plant roots. And this explains why the application of humic substances like humic and fulvic acids can show inconsistent, but generally positive results on plant growth. The variability can be due to the source, the environmental conditions when they were applied, and actually to the particular crop they're being used on. Amino acids, they're obtained by chemical and enzymatic protein hydrolysis from both plant and animal products. Direct effects on plants can include modulation of nitrogen uptake and assimilation. Chelating effects are also reported that contribute to micronutrient mobility and uptake. And indirect effects on plant nutrition and growth have been seen as these proteins are known to increase microbial biomass, soil respiration, and overall soil fertility. Again, variable, but in many cases, significant improvements in yield and quality traits have been seen in agricultural and horticultural crops. So hopefully that's given a bit of an overview about why we're talking about biostimulants today, what are the different types. Clearly, it's a complex subject because we're talking about so much interaction about things that are going on in the soil and the plant. So I'll hand over to Antonis, and hopefully he can continue the conversation on our discussion today.
Thank you, Chris. Thank you very much. So, starting from where you, let's say, you stopped, of course, you explained in a very good way that when we talk about biostimulants, there are many different ingredients that we can use. You explained very well that there are non-microbial biostimulants, microbial biostimulants, and even combinations of these raw materials for developing a product. Also, at the same time, in order to add further on the complexity, we can have different extraction methods. So when we talk about, for example, amino acids, you said that we can use either enzymatic hydrolysis or acid or alkaline hydrolysis. The same can apply for other categories of products like the seaweeds, where we can have seaweeds based on acid hydrolysis and alkaline hydrolysis, or even using, let's say, other methods of extraction without using any chemical. and the same can apply for the rest of the raw materials. And on top of that, we have different formulations, either solutions, suspension concentrates, we can have powder, solids, and of course the combination of different ingredients in a formulation that can make even more, let's say, confusing the whole environment of biostimulants. So out there, probably, indeed, you see the UK market and also the European market or the global market, there are, I would say, hundreds or even thousands of products, and it's very difficult for the end user to decide, okay, which one should I use? And what is the best solution for me to use for my crop, for the problem that I want to address? And this is what we want to discuss a little bit more today on what is, at least from a manufacturer point of view, what should be the appropriate process to follow in order to develop a formulation that will be a unique formulation and an effective formulation to be used by the end users in order to deliver specific effects, specific claims, because this is something that is very important. And we saw also from the responses from the polls that many of the end users are using biostimulants either for nutrient use efficiency, which I think scored the highest level, and the second category was abiotic stress tolerance, and the third one was crop quality. So we can already see that the users of biostimulants here in the UK, they already have an understanding that they should use a specific product for a specific function, which for me, it's a very good starting point to know when and why to use a biostimulant product. But let's explain a little bit more what are the main, let's say, parameters that we need to take into consideration when we are developing a biostimulant or when the end user wants to select the appropriate product to use for his crop. And of course, the first thing that we need to mention is that it's very important that the combination of different ingredients, we mentioned before the different ingredients, the combination of different ingredients is what it matters when we talk about the product development of a biostimulant product. And why we say this? As you can see also in the slide, I tried to bring an example using the Lego. And for those of you that are, let's say, fans of Lego, you might know the game with the duck. We have a group of people that can have the same 6 pieces, 6 building blocks of Lego, and they are asked to develop, to build a duck. And as you can see in this slide, we asked from our colleagues earlier today, 6 colleagues, to do this, and each one of them built a completely different duck. It's exactly the same, the analogy is exactly the same with the biostimulants. All the companies can have the same raw materials, they can have the same ingredients to be used for a product, but this does not mean that all the companies will develop exactly the same formulation. This will depend on the experience, the knowledge, the know-how that the company has, the expertise that the company has on the R&D department, on the chemical development department. And this is very important in order to be able to combine in the best way the active ingredients and the other additives in order to develop a unique and effective formulation, a stable formulation, and of course a formulation that is safe for the crops. And this is also where we as Yara, having also the expertise and the know-how from more than 50 years of experience working with the Yara Vita portfolio, that we are utilizing this knowledge in order to develop also our biostimulant portfolio. But of course, talking about the combination of the ingredients in a formulation is just one of the, let's say, the parameters that we need to take into consideration. In addition to that, and as we said before, it's very important, what are the expected effects for the specific formulation. And as we saw in the poll, our participants, they are very well aware that they need to use specific products for specific reasons. So as you very well explained earlier in your previous slides, when we talk about the product development of biostimulants, first we need to identify what kind of effect we would like our product to have. Is it a product that would like to have an effect on enhancing tolerance to drought? In that case, we need to focus more on abiotic stress tolerance and search to find the compounds that will work in that direction. If we want, for example, to have a product that will help enhancing nutrient use efficiency, either nitrogen fixation, phosphorus solubilization, or any other form of nutrient use efficiency and uptake, then again, we need to search on another, let's say, pool of ingredients in order to select the appropriate ones that could be used for the formulation. And of course, talking about crop quality and yield increase, again, it's the same concept. You need to identify what are the molecules, what are the the ingredients that you need to use in that direction, having always in mind that the effect is what it matters. The effect of the product on specific crops is what drives the product development for a biostimulant product. And of course, talking about having, of course, it's important to have high quality raw materials, having a formulation that combines different active ingredients in order to enhance the efficacy of the formulation and deliver specific effects. But of course, there is another, let's say, important parameter that we need to validate these effects. And this is something that it's not something that you can do easily in, let's say, in a month or two. And requires quite a lot of effort, requires a lot of trials, both greenhouse trials, but also a lot of open field trials in order to be in position to validate the timings, the rates of application, the crops that you are going to use the product, and be able to understand the mode of action of your product and be able to give the best, let's say, recommendation in terms of product position. So this is the, let's say, the three main parameters, factors that we need to take into consideration when we are talking about development of a biostimulant product. Formulation, combination of the different active ingredients, what is the specific effects that we want to deliver, and of course, how we validate this effect, the application rates and the timings. All these are very important for us to be able to, as a company at least, to be able to give a proper recommendation and be confident that what we are commercializing as a solution will deliver the expected benefits to the farmers. And of course, one thing that we need to highlight is biostimulants is a different category of products. They are not, let's say, like the plant protection products. They are not like the fertilizers. So we cannot expect to have a similar behavior as the behavior of a crop protection product. So also when we talk about in terms of efficacy, in terms of consistency of this kind of products, it's not the same level as it will be, for example, with an insecticide or a fungicide, where we expect to have a minimum of 90–95% efficacy and consistency in order to be able, let's say, to have a successful formulation. Even at European level with the new regulation, the requirements for, of course, there are requirements for trial for biostimulants, but we don't have the same level, let's say, of requirement as with the crop protection products. And as we can see, and this is the experience at least from our biostimulants, our products, and as you can see in this slide, when we compile a database of more than 300 scientific trials, we can see that we can talk about a win rate of around 75%. So having a biostimulant product that will have a win rate around 75% is considered to be, let's say, significant, and it can be, let's say, in terms of efficacy of the formulation at a very high level. But we cannot expect having a biostimulant that will consistently, at 100% of the cases, deliver a positive effect, because this is something that even with fertilizers we don't see. Even with other product categories, we don't see, let's say, 100% consistency of the efficacy of a product. And this is something that we need to have in our mind. I remember one of the questions that we received before the webinar that was asking about this, and that's why I wanted to mention this. But let's explain a little bit more the whole process on the product development, because this is what we are supposed to discuss today in this webinar as part of this discussion. And of course, for us as a company, we have already established a systematic process on our product development, starting from the ideation process, where, as we said before, we will get the input from the markets, from our R&D team, from the product management team, in order to say, I would like to develop a biostimulant that will have a specific effect. This is how we start with all our projects. And then this will be given to the technical committee to be evaluated, and then the R&D and the chemical development team, they will start the scouting process, the product development process, and as soon as we have the final formulation, of course, the next step for us is to see from an industrial production point of view, if this can be, let's say, something to be produced at large scale. And before going to the commercialization stage, of course, we need to do the registration of the products in the relevant markets, following the relevant regulations for markets. But for me, in order to focus a little bit more on the most important part, on the product development stage, I have another slide where we can go into more details and explain what are the steps that we are following in order to develop a biostimulant product. And to see that this is a process that is quite, let's say, exhaustive as a process, and is a process that could last between 3 and 5 years in order to deliver a new biostimulant product in the market. So starting, as we said before, the first step is the idea. We have an idea to develop a biostimulant, let's say, for example, that is going to have an effect on nitrogen user efficiency. In that case, the first thing that we are going to do, our R&D and chemical development teams are going to start scouting the literature in order to see which are the potential bioactive compounds, ingredients, and even nutrients that we can use in order to deliver the effect on nitrogen use efficiency. After having, let's say, the literature search, and after having, let's say, a list of components that I have seen some of the list from our R&D team that could enlist even more than 100 components initially. So they have then to see which of them can be registered, which of them are safe for the end user. So there are many parameters that we are checking during the scouting phase in order to make sure that we are going to use components that are safe for the end user, but also for the consumer later on. And then we are going to list them based also on the potential effect that they have for the specific claim that we want to have for the product. As soon as we have, let's say, a short list of ingredients, then the chemical development team will start working, play, as I like to say, with different prototypes. So they will start developing different prototypes with different active ingredients, different combinations, different ratios of the active ingredients. And then it's up to the R&D team to start doing the initial screening of these prototypes with, let's say, bioassays or lab studies in order to understand which of these prototypes are performing better. So having this process that could take quite significant amount of time, I can tell you that recently we launched a product internally, and the chemical development team had developed more than 100 prototypes before reaching to the final formulation. So even the chemical development process could be, let's say, quite extensive, because it requires a lot of tweaks in order to have an effective formulation, a stable formulation, safe formulation for the crop. There are many parameters that we need to take into consideration, and of course, at the same time, to have a long, let's say, shelf life for the formulation. So if we are lucky and we have, let's say, good results on the biological screening phase, then the 2 or 3 most, let's say, potent prototypes, the most effective ones, will be then transferred to the next stage, which is the small scale evaluation. At this stage, we are starting doing studies under controlled environment. For example, if it is a product that we talk about enhancing tolerance to abiotic stress, of course, we need to test the product under different conditions of stress. Drought stress, heat stress, and see how the product is performing, depending, of course, on the target that we have as an effect for the product. So this is a period where we will also define what are the application methods for the product, what are the application timings, and the range of rates that the product can be used in the different crops. And of course, having this information in this stage is very critical in order for us to go to the next stage, that is the stage of the field development, where we want to validate, again, the crops, the application times, and the application rates, but this time, this will be done in open field crops. So we need to do this also at a bigger scale, and this is something that we need to do under various conditions. So we are going to run trials not only for 1 season or in 1 region, but we are going to run trials for at least 2 seasons and globally. At least as Yara, we are likely to have, let's say, the footprint as a company to be able to work at the global scale, and we are able to test our products not only in UK, where it's our main, let's say, where our chemical development team is based, but we can test our products in Europe, in Asia, in Latin America, and North America, and be able to understand how our formulations are working under different conditions. Because it's very important to highlight that when we talk about the efficacy of biostimulants, this will be also affected by the soil parameters, by the climate conditions, the crops, even sometimes, even the varieties can make a difference of the crop varieties. So for us, it's very important to understand the effect of our formulations, not only in one country or one type of conditions, but try, let's say, to have a more, let's say, broad idea how our product is working under different kind of conditions and in different markets. And as I mentioned in the beginning, this whole process for us could take from 3 to 5 years from the ideation process until we have the green light from the R&D that we can commercialize this formulation. And this is very important because having all this background, let's say, knowledge helps a lot to understand how to recommend the product and under what conditions we can use, let's say, the product. And of course, just for me to explain a little bit more in details, when we talk about the evaluation phase, either the bioassays, the lab studies, or the small-scale studies, we have already our facilities. We have in our R&D headquarters, I would say, in Germany, we have a dedicated facility where we are working for the development of biostimulants, where we have also a diverse team of scientists. It's very important because in our team, and I know it's not only in Yara, but also in other companies, that when we talk about the R&D, we don't just have, for example, plant physiologists, but in our team, it is consisted by plant physiologists, molecular biologists, agronomists, biochemists, soil scientists. You need to have different, let's say, expertise, because each one of them will contribute with the knowledge that it has in order to develop the formulation and also to evaluate the formulation in the best way. And of course, having the appropriate equipment to do this is also something very, very important. And of course, we as a company, we are investing in that direction. But you can understand from what we explained until now that the process of development biostimulant product is not just, let's get seaweed, add it in the water, mix it with the water, put it in a bottle and sell it. It's a rigorous process. It's a process that has a lot of requirements, a lot of steps in order to be able to understand how the product is working, under what kind of conditions it's working. And I'm happy to say that we see more and more now companies in this sector to follow a similar process. That means that more and more we will have products in the market of high quality and having a higher level of consistency in terms of the performance of this product in the market. And this is, of course, where we also as a company have already worked in order to develop a portfolio that reflects this product development process, this concept of having specific products for specific effects. So we don't want just to have a biostimulant product that is appropriate for everything, because we know that it's not the one size fits all.
No.
In the case of biostimulants, depending on the claim, depending on the effect that you want to have, you need to select the appropriate product, or depending on the application method. Do you want the product to be used as a fertilizer coating in order to enhance nutrient use efficiency of your fertilizer? Then you should select the appropriate product that was developed for this specific application. If it is for a seed treatment, it will be another solution that was developed exactly to be used as a seed treatment, because we need to take into consideration also some other parameters. For example, on the seed, we need to make sure that we are not affecting the seed germination, the seed viability, which you cannot, let's say, ensure if you don't have a dedicated product developed for this specific application. And of course, when we talk about foliar or fertigation biostimulants, again, you should select the product that was developed for this specific applications, because if it is a foliar product, it will contain also some additional components that will further enhance the uptake of the bioactive components, and when you use it in fertigation, again, you will take into consideration some other parameters. So all of these are very important to be taken into consideration when you are developing a formulation. It's not an easy thing to do, and it requires quite a lot of time in order to deliver an effective formulation in the market. So for me, I tried in this short, let's say, short period of time to give an overview of what are the steps on developing a biostimulant product and what are the main, let's say, parameters that we need to take into consideration. So again, we talk about combination of different ingredients with the objective of delivering a specific effect on the crops. And of course, we need always to validate this effect based on science, based on trials that will help us to understand the mode of action and how the product is performing under different conditions. So that was from my side. I hope that I gave, let's say, some more clarity on how we work on developing biostimulants, and I will give the floor to you back, Chris, in order to say what is the next step now.
Yeah. No, thank you, Antonis. That was very interesting. Hopefully you found that helpful. As I said, today's webinar, the intention was really just to take it as an introduction, if you like. It's a big, complex subject, as I've said more than once now. But the intention is, after today, is to have a second webinar. That's scheduled for 2 o'clock on Monday, the 19th of February. I hope if you've enjoyed today's webinar, you'll be able to join us for that. And in that one, we'll build on what we've talked about today, myself and Antonis, and we'll look at in a bit more detail on how Yara has developed those products and some of the specific crop situations where we've seen positive results from using biostimulant based products. Thank you very much, and thank you to Antonis.
You are welcome, Chris. Thank you for the invitation.
Thank you. Bye-bye.